Today we're going to look at germination and the control of germination. Before we do that, we need to look at the structure of a seed. So we take a badly drawn cross section of a seed. You're going to have a small hole, very important. Beside that, you're going to have something that looks like a leaf and another piece. And all the way around the outside, you're going to have a protective layer. To label this, the protective coat is called a tester. The bit that looks like a root is called a radicle. The bit that looks like a leaf is called a plumule. These two together make up the embryo, the young developing plant. And the rest of the seed, the other 95% of it, is the food store or cotyledon. This is the structure of a typical plant. You can quickly see when you look at this just how much of this plant is actually food store. You can see how much of the seed is food store. All the green here, shouldn't use green really, it'll mix us up with chlorophyll, is food store. This is why seeds can last for many years. Underground, in the forest, on the ground, anywhere. There's a food store and it's being used very slowly. That is our starting point for germination. There are two levels we can look at germination from. The first level we can look at is the physical level. This is where we look at what actually happens. The first step on a physical level is that the tester splits. The tester being the seed coat. Now there's a variety of causes of this, but ultimately water swells up the inside of the seed, the cotyledon, which puts pressure on to split the tester. The root grows out from the radicle, or the radicle grows up and out. This is bizarre. Why would a root grow up? If we take our badly drawn seed, the radicle comes up, grows up, around, and then down. And it'll continue to grow down towards water. After this, the root straightens up, which has the effect of lifting the seed upwards. So if you can imagine, it straightens, lifting this up. So we end up with something looking like this. Never seen a blue seed before? Here we go, it's lifted up, and we've got our seed here. It's been elevated. Finally, we have the shoot grows out. So we end up with something that looks like, and this is where we can tell, is our plant a monocot, which would have one first leaf, usually parallel veins, or is our plant a dicot, which would generally have two leaves at the beginning with non-parallel leaf veins. So as a reminder, monocot, one emerging leaf, one cotyledon, which is the food store, parallel veins in the leaf, and fibrous roots. Well, a dicot is going to start with two initial leaves. They're going to have web-like veins on the leaf, not parallel. They're going to have branching roots, not just running straight off the stem, they're going to fork and branch. So this is the physical level of what's happening when a plant seed germinates. However, Ivy is more interested in chemicals. What's causing this to happen? So we can also look at this at a chemical level, which is really what they're after when you have a question on control of germination. The first step, water diffuses, science word, in, which causes gibberellin, or actually gibberellins, it's a group of hormones, to hydrate. When they're dry, they have no effect, they do nothing. Once they hydrate, the gibberellin causes the production of amylase, which is the enzyme that converts starch in the seed into maltose. Maltose, of course, being a small soluble sugar, which is one of the energy stores for the developing embryo. So the maltose, once it is created, is small enough to be water soluble, and it can diffuse towards the embryo, where it is an energy source. Here's our first step, chemical level. Following this, we have step 2, where water keeps on diffusing in until the pressure of the enlarged cotyledon, or energy store, causes the tester, or seed coat, to split. Now once the tester is split, this leads to more oxygen and water diffusing in. And of course, once we've got a lot more oxygen coming in, it will lead to aerobic respiration, 19 times more energy. Eventually a plant is going to be big enough to break the surface, to have leaves out, to then start using the energy from the sun, the light, to produce its own food. It will no longer be reliant on the food stores inside the seed, or inside the cotyledon. One more thing to mention is that at some point, with water also in there, our proteins inside the cotyledon, the proteins inside the cotyledon will be hydrolyzed. Hydrolyzed, look up hydrolysis, water splitting, opposite of condensation, into amino acids, which are then used as the building blocks for the developing plant. Until we get to the sun and can create our own, we have to have these ready to go and provided.